Welcome back to my YouTube channel Bio Shastry. Nodi Kali Kelitali. This is myself Soudhaminishree Shastry yet with another video on how to write precisely in biology practical examination to score 30 on 30. So before going deep into the topic, those who are new to my channel, please do subscribe my channel. Yes, let's get started. So in my previous video, I already have told you that in the biology practical examination, you will not be provided with the additional sheet. So you have to manage your writing skills and you should be very precise, up to the point, be specific and write how much it is necessary. Clear? So the first thing is you have to divide your answer sheet. I'm just taken one rough sheet for you guys, so how you can manage your answer paper. Okay, so this is how it looks. Your answer paper, you will be having there will be a column to write the registration number in all the 4 pages. You have to write the registration number in all the 4 pages. There won't be given any extra sheet for you to write. Okay, so the first page you have to divide this. You have to put the margin, you have to write the question number and you have to write the question number is A. I already told you the question number means it will be on the A. So the A question will be on the lottery system. You will be having 4 choices, that is either the TS of ovary, nucleus staining, pollen tube growth on stigma or pollen germination. So the 4 questions you will be having. I'll show you how much to write and how to write. Okay, the entire sheet you will be using for part A, that is or the question number 1A. Then turn the page. In the second page you will be dividing the page for the 2 questions, that is the second question B. This is for the mitosis. You have to identify the slide which phase you will be having in your prepared slide. Then around 10 lines from the down you count and write the third question C. This is for your pH question. Then go to the third page, you will be having the fourth question D. You have to identify and you have to give the reasons which they have kept, either the TS of ovary they have kept or the mammalian blastula or VS of blastula. You have to write the TS, VS properly over here in this question. Then the E question you will write around 8 to 9 lines from the down you count and you mark it as 5 and write E. So here you have to write only 2 lines, that is which the organism you have identified and what disease it will cause. Okay, so I hope the second page is for 2 questions and in the third page you have 4th question and the 5th question, and in the last page you will be writing 6F. So probably under F the experiments what they will be keeping is either the photograph or the specimens for the xeric, hydric adaptations of the animals and the plants. Okay, and also there will be the diagrams or the photographs for the homologous and analogous structures. So you just leave this space rough. Okay, I hope how you have to divide your 4 pages in your answer sheet and you have to maintain this space. After each question you draw the line, it will be beautiful so that there is no mixing up of the question and you can't repeat the question and there is no room for you to scratch and write and there won't be no additional sheet for you guys. Okay, so you have to finish your entire examination in the 4 pages. So I hope this much is clear. In all the pages you will be having a column for you to write the registration number. You have to write in all the 4 pages. Okay. I told you regarding the 1A, you will be having a lottery system. Okay, for each of the experiments I will be showing how much you have to write for your exam. So here the marks breakup will be for your preparation of the slide. You have to take the TS of ovary and you have to submit, and for the writing part you have to write. So probably the flower will be given here will be your hibiscus. For the hibiscus I am showing you, you have to write the result, you have to underline. The number of locules in ovary is 5, that is pentalocular. This word you have to mention. And the type of placentation is axile. So if you write this much, this is more sufficient. Okay. So if you get the question on the pollen tube growth on stigma, you have to draw the labeled diagram. You have to prepare a slide, and your slide should show all the details how the pollen has germinated. You have to mention the pollen grain, pollen tube, then the style, and you have to write a footnote, that is growth of pollen tube through the style. So you have to write the footnote as well. So this is for the experiment pollen tube growth on stigma. For the nuclear staining, I told you for the nuclear staining, the specimen will be given will be your onion peel. So you have to stain it with acetocarmine, prepare the slide, show for your examiner, and for writing part you have to write result. The shape of the cell is rectangular. The number of nuclei is 1. So either you can write this, or I suggest you, if you write the second version, that is the number of nuclei per cell is 1. If you write this sentence, it will be more appropriate. This much you have to write for your experiment nuclear staining. In the lottery system, if you get the question A as pollen germination experiment, you have to take the observation table like this. If there is only one observation, no need to write the average. If it is more than one trial, that is 1, 2, 3, you have taken, then you have to take the average of all these things. Okay. So in the second column, you have to mention total number of pollens you will get in the microscopic field, that is capital N, and total number of pollen germinated, that is small n, and you have to calculate the percentage of pollen germination by using a formula small n into 100 divided by n. So this is a percentage of pollen germination, n into 100 by n you have to mention, and if you have taken a 3 trials, you have to write the average of the small n, capital N, and then you have to substitute here in this formula, and at the end you have to write the result. The percentage of pollen germination is dash %. So you have to mention this %, and this line is very essential for reporting how much % it is there. You will be having 1 mark over here. So this line is important, and you should not forget this percentage symbol. As earlier I told you, in the second page you will be writing, again you have to write your registration number, that box will be given. The second question, that is B, that is mitosis. You have to write the stage is identified as dash. So in the dash you will be writing either you saw the prophase or the metaphase or the anaphase or the telophase, and you have to write the labeled diagram. Why I have given the box is, this box refers the cell. Within the cell you have to show either of the stages here, and you have to write the footnote whether it was a prophase or it was a metaphase or it was a anaphase or a telophase. So within the cell you have to show the stages. So if you write this much, you will be having a marks for your identification 1 mark, and you will be having another marks for your labeled diagram. Okay. Then for the third question, you have to make a tabular column for the pH, that is the sample you have to take as a C1 and C2, because in the examiner they will be labeled as either C1, C2. Okay. For the C1, what was the pH? You have to write over here. For the C2, what was there in your broad range paper, narrow range paper, or in the universal pH solution, how much you got, and you have to mention the result. What is the result? If you get the pH value from 1 to 6, that is acidic. If you get the 7, that is neutral, and above 7, that is 8 to 14, if you get, that is a basic. So you have to write the result as the pH of C1 sample is dash. Suppose if you get 1, write it 1, and it is dash in nature. Here in the dash you have to write acidic. Since it is 1, it is acidic. So the pH of C2 sample is 7. 7 means it is neutral. So 7, and it is neutral in nature. So this is how you are going to write the result for your C. Isn't it easy to write? Next, in the third page you will be writing the question 4 and question 5. For the question 4, you will be having the slide or photograph whatever it has kept, you have to write. If they have focused the slide under a microscope, you have to write the slide is identified as dash. For example, it can be TS of ovary or it can be TS of testes, VS of ovary showing the female gametophyte, or the TS of blastula. For this identification, mentioning the TS and VS is very important. If you write just the ovary, you will lose the marks. You have to write TS of ovary. Suppose if they have kept the photograph, don't write as slide. Use your common sense. The particular photograph D is identified as dash, and you have to write the 2 reasons. Only 2 valid reasons which is given in your lab manual. You read it, and why did you identify this slide? What was the key feature did you identify in the particular slide? You will be writing the 2 points. Okay. Then in the E question, that is 5th question, the specimen will be kept or the photograph will be kept for the disease causing organism. So how you will be writing is, the disease causing organism is dash and it causes dash disease. So you have to underline this like this. So in the fill in the blank, you have to fill the blank as which was the organism did you identify and what it causes. There are only 4 organisms in your practical. Entamoeba histolytica. So here you have to use the binomial nomenclature for writing the scientific name. The first word you will be writing with the capital letter and the second word you will be writing the small letter. Entamoeba histolytica is the disease causing organism and it causes amoebic dysentery or the amoebiasis. Or you might have got the Plasmodium vivox as the disease causing organism. Then in that case you have to write the disease as malaria fever. If you got the Ascaris lumbricoides, the disease caused by the Ascaris is Ascariasis. Suppose you might get the Trichophyton rubrum as a disease causing organism in your exam, then you have to write as ringworm. I have shown all the disease causing organism and what disease it will cause. If you practice this, you have to just identify, and this much is the writing part in your examination. No need to write all the 4. Whichever one you have got in the exam, you have to write the disease causing organism is dash and it causes dash disease. And in the last 4th page, you will be writing the question 6, mention as if the specimen or photograph or the model is identified as. Don't write all the 3 words, whether they have kept the specimen, they have kept the photograph or the model. You please check in your examination, whichever is there, you mention that word and identified as. And mentioning the words like adaptation in plants, either the homologous structure or the analogous structure, this word is highly essential. And when you are writing the reasons, you have to write the 2 reasons. Don't give the numbering from here. That's why I have caught here, because this left hand side of your margin indicates the question number of your question paper. So if you are giving the points, you have to give inside the margin and draw the line after completion of your question. So only this much thing you have to write in your examination. I hope I shown you the paper how you could write precisely in the answer sheet without taking additional sheet for your examination. And also students, remember, there will be no extra sheets will be provided for your practical examination. So make a mindset and be clear in which page you are going to write how much, and if you practice this and if you go to the examination, definitely it will be helping for you guys. I hope this video was useful for you, and I wish I could make easy for you to understand how you can write precisely, specifically and up to the point for the examination, and it will definitely going to help you to score better marks in your practical examination biology. Thank you for watching the video till the end. I hope this video was useful for you. Please like the video, and those who are not yet subscribed to my channel, please do subscribe my channel Bayush Shastry so that you don't miss any informative videos on the board examination. And share this content with your PU friends so that it will be helpful for them to score better marks. Thank you.